Okay, our next speaker and last speaker of the day is Dr. Barbara Sorkin. Dr. Sorkin is also part of the Office of Dietary Supplements. She is the director of the NIH Centers for Advancing Research on Botanicals and Other Natural Products program, and we are very happy to have her here today to discuss the challenges in research on botanical dietary supplements.
Thank you, Luisa. Thank you all for being here today and for your interest in dietary supplements research. As Luisa said, I actually co-direct the Botanical Research Center's program at NIH, which has been subsumed into the somewhat larger Carbon program, the Centers for Advancing Research on Botanicals and Other Natural Products, or Carbon. I co-direct that. Paul Coates probably told you this morning that most of what the Office of Dietary Supplements do, we do in collaboration with other agencies, NIH components, et cetera. And the Carbon program, the Botanical Research Centers, is an example of that. Dr. Ursha told you that we collaborate quite a bit with our colleagues at the National Center for Complementary and Integrative Health, or NCCIH, and this is a collaboration with them. And I'm going to talk about it a little bit at the end of this talk. So I think this is the first talk that you've had today that's fully focused on research as opposed to regulation, a word that I may not actually use again unless I have a question about it at the end. And you're going to have other talks over the next day and a half about research. So what I want to do is use this talk to talk a little bit about some of the concepts, things to think about if you're designing research and doing research, and things that you should think about equally much if you're reading research and assessing what kind of weight to give the research that you're looking at. So we do a lot of biomedical research here at NIH. Why do we do it? What are we trying to get out of that research? It's a chunk of taxpayer money, about $30 billion a year. We're trying to improve health outcomes. And in order for the research to improve health outcomes, it needs to be reproducible, right? Because otherwise, as Joe was talking about with the Cochrane reviews, otherwise, if we keep getting different results, we don't know what it means and we don't know what to build on and how to build on it. And I'm going to hope that you will take with you to Lebanese taberna, between which and now I am standing. Great grammar there. Three things that I'd like you to take with you. One, that in order for us to get the biggest bang for our buck when we do this research, in order for us to get the most out of the time that we, and if it's a human study, our participants put into this research, we need to understand, in order to understand the health effects, we need to know what's in the bottle, as we've been discussing, but we need to understand causal molecular mechanisms as much as possible in order to get the most out of it. And I'll be talking about this more later. Understanding causal molecular mechanisms, point two, as Adam and others have said, these are complex, and especially for the botanicals, which is why they're additionally challenging, they're inherently variable. So we probably need to think about more than a single marker compound. We probably need to understand the chemistry and the effects of the chemistry on the biology at a more detailed molecular level in order for that research to be reproducible enough for us to build a better understanding of what these things mean for public health. And point three will come back to me when I get to it, I guess. So we want reproducible research. If it's not, it doesn't help us. Why botanicals if they're so complex and inherently variable and the Cochrane reviews are all over the place? For one thing, 1 in 6 Americans uses a botanical dietary supplement. For another, we know that there are bioactives in there. This is a partial list of some of the drugs in our current pharmacopoeia that originate from botanicals. And last but not least, we're still trying to understand what it is about fruits and vegetables. Is it the polyphenols that are important for health? And if so, how? Well, I argue that understanding the mechanisms of action and the actives that are less common than the polyphenols, things like hypericin or hyperforin in St. John's wort that Joe mentioned quite a bit and Adam also showed you. Those are not going to have as much confounding from background, from diet. So they may be easier to figure out, and then we may be able to take that same paradigm that we use for understanding how those bioactives exert their bioactivities. Not that that's always easy, as you'll see. We may be able to take that back to the more common phytochemicals and apply it to understanding how those work. So here's the outline for today. We've talked about the goal. I'm going to spend most of the talk on some of the challenges that are not non-existent for other dietary supplements, but I think greater because of the complexity and variability of botanicals. Because this is the first really research-focused talk, I want to say a few general words about research challenges to going from the lab to, this was the third point, really thinking about the research. How does the question that you're asking in the lab, how does your experimental design relate to the question you're really interested in? Whether that's what this product means for your personal health or public health in general, how to generalize it, I think requires that you really think about what you're doing and what your options are and why you're choosing what you're choosing. And lastly, I will talk about the carbon centers and what NIH is doing to help you do rigorous research on dietary supplements, especially botanicals. So products, stronger in botanicals, these are some of the issues I'm going to take you through. We'll start with source identification. And as Adam pointed out, macroscopic morphological identification is a commonly used, it's a traditional method. So how many of you can tell me what this traditionally used flavoring is? Star anise. Well, that's a good guess, but it could also be this. They look alike, but okay, so chemistry is really important, right? Because presumably what's toxic in there is the chemical. I'm told that experienced botanists can tell these apart, but so this brings us back a little bit, and we'll get there in a minute, to a phrase that Adam used, orthogonal methods. One method that you could use to distinguish between related species is DNA barcoding. I don't want to go through this in any detail. I simply want to point out that most of the time when you're thinking about biological activity of botanicals, you're thinking about chemicals that are fairly unique to the plant, which DNA is not. You don't think of the DNA sequence as being responsible for the activity. So the DNA sequence is going to be the same whether you're looking at the aerial parts or the root, but the chemistry might be quite different. Not to mention that the chemistry is going to vary from season to season and altitude to altitude. So DNA sequence for regulatory purposes to see whether you have the expected species may sometimes be useful. If you want to ask me about pitfalls, there are a lot listed on the slide. We can go back to that later. I don't want to take the time now. But clearly, chemistry has more relevance to the question you're really asking, which is what is the biological effect of this plant? The species, of course, is going to constrain the chemistry, but it's not going to define the chemistry. So orthogonal methods, methods that are based on different scientific principles. If you get a result in the lab that's really, really important, like you're going to give this to study participants and you want to make sure that you don't poison them, or you get a result that's really surprising, or you see a published result that's really surprising, one thing to look for is, did they use two methods based on different scientific principles? I'll come back to this later with one of the carbon centers that's using orthogonal methods to generate hypotheses about mechanisms of action. But for example, if you were doing DNA barcoding of a product and you used two different primer sets, would that be orthogonal methods? No. If, for example, you had made an extract that didn't include any DNA from your St. John's wort, which might be a perfectly good way to get the hypericin and the hyperforin out of the St. John's wort, but not the DNA, it wouldn't pick up any St. John's wort species sequence, which wouldn't mean it wasn't a good extract. And it wouldn't matter what primer set you used, it still wouldn't pick up any signal from the St. John's wort. But if you had a rice excipient in there, it might pick up the rice. So using two different primer sets is not orthogonal. They're both based on the same principal DNA sequence. So orthogonal method would be using a chemical method, or macroscopic identification if you had pieces of the plant, or doing a bioassay. Does it have an effect on animal behavior or central nervous system activity? So since I'm talking about St. Johns Wort, what's so complicated about looking at the chemistry? Well, as Joe said, he pointed out that the Cochrane reviews in many cases say that there's no definitive data for bioactivity of botanicals. I would add on to that for most botanicals, we don't have definitive information on what the bioactives are, which makes it a little bit hard to quantitate the bioactive as a way of making sure that you've got a reproducible research material for your biological study. Well, okay, so could you just standardize to a marker compound, like hypericin? Can I use this? No. Oh, I can. No, you can't see it there. Okay. Hypericin or hyperforin. Could you just say, okay, my sample is going to be between 0.1 and 0.5% hypericin, which looks like that, and it's going to have certain ratios to all the other compounds. The other reason I put this up here for you is to point out, Adam said these things were complex, Joe said they were complex. This gives you a sense of the complexity and also of the extent to which we don't know what's in there. That there are unknown unknowns that could be influencing bioactivity. So more than a third of these typical extracts is what the chemists call structurally unassigned, which I would translate into English as there's not enough of it for us to figure out what the chemical structure is. Is that reasonable, Joe? So you've got these complex things, and not only are they complex, but the ratios of the different components are not particularly predictable. So these are data from one of the botanical research centers, the one at University of Illinois, Chicago. They look at botanicals that are commonly used by perimenopausal and postmenopausal women. And these are samples from a fraction that has estrogen receptor-related activity from Angelica sinensis, or Dongquai. That one is a marker compound. And what they're doing here in this chart is they're looking at, in about 20, I didn't count the bars, different batches of Dongquai extract. They've normalized, they've said, okay, these 6 components that are in our bioactive fraction, we're going to set them to 100%, and then we're going to use standardized methods to look at the proportions of them in the different preparations. And you can see that it varies all over the place. So clearly what you want to be reporting when you're reporting research, if you want other people to be able to build on it, if you want to be able to repeat it in your own lab with the next batch of Angelica sinensis extract, is you want to characterize it to an extent that's determined really by your understanding of how the chemistry affects the biology that you're interested in. So here they have 6 components. One thing that it would be really interesting to do that I hope these guys are doing is looking at how the different ratios of these components in the bioactive fraction affect the bioactivity, because I think you would learn something from that about what their different individual contributions are. So why focus on multiple components? What's the evidence that, in fact, multiple components contribute to the bioactivity of botanicals? Let me give you one more example. This is from traditionally used botanical for fever, traditional Asian medicinal systems, Artemisia annua. 2UU shared the Nobel Prize in 2015 for the isolation of artemisinin. Dihydroartemisinin combination therapy is the World Health Organization current frontline drug for malaria. And what you're seeing here is comparison of a whole plant Artemisia annua preparation and its effect on malaria parasitemia in a mouse model. You can see parasitemia coming up. A single dose is given of either the whole plant or an equivalent amount of artemisinin. And you can see that there are clearly other things contributing to the activity of the artemisinin in the whole plant preparation that are not artemisinin. So there again, there are other things contributing. There are many ways that those other things could be contributing to activity, and I'm happy to answer questions about that at the end, hint. So the importance of looking at multiple components in botanical extracts, and of doing that in an informed way, because it's really expensive to look at every single component. So stability. Obviously, to the extent that you know what you need for bioactivity, you know how to look at stability. Going back to our St. Johns Wort extract, and giving you an example of why understanding molecular causal mechanism of action and why looking at stability are really helpful in getting the most out of a study. So this is a St. Johns Wort study. It was published in JAMA, and it's in your reference list, but JAMA 2008. They were looking at, and this is a pilot study, they looked in 58 participants, oh, that works. 58 participants, 27 per group. They got 900 milligrams a day of a 0.3% hypericin preparation of St. Johns Wort. They were hoping that it would have some effect on symptoms of ADHD, and you can see that at the end of 8 weeks, it had absolutely no effect. They completely failed to disprove the null. Now, they analyzed their product at the beginning of the study. It was indeed at that point 0.3% hypericin, so they had sort of done the right thing, but when they analyzed it at the end of the study, it was down to 0.13%. Beyond that, by the time they finished the study, and obviously 60 participants, 8 weeks each, it took more than 8 weeks total. At that point, there were reports in the literature that the bioavailability, this is really important, bioavailable levels of hypericin in the serum were probably not enough to have central nervous system activity, to have mental health activity. So people were thinking that it might be the hyperforin, but in order to have an effect on mental health, people were thinking that you needed to have at least 3% hyperforin, which obviously they did not have. So what have they learned from this study? Well, maybe they've learned that next time you look at stability of your product before you start the study and make sure that it's going to stay, or you monitor it during the study. But how might they have learned more? Thinking positively going forward, how would you design a study so that you would get as much out of it as you can? What would you like to know going into it? Hint, hint, causal molecular mechanisms. So in my beautiful schematic PowerPoint drawing here, I'm really good at this, what I'm saying is that to the extent possible, you would like to have good evidence for which of the components in the plant, this is a leaf, you can tell, which are the bioactives, which ones should you be looking at, what should you standardize on, what should you be looking at stability of, so that if you fail to, you stop the experiment as soon as your product falls below your specification. You make sure that it's going to meet your specifications throughout the study. And then it would be really nice to have good evidence for where it needs to get to in the body. Do you need a certain level in the serum? Do you need it to interact with the cell membrane, with specific membrane receptors, with some protein inside the cell? Because then you would be able to monitor, do you have enough bioavailability? And in fact, is the input compound your active, or is it a metabolite? So that would all be nice stuff to know, because then if you don't see an effect at the end, you could look and see, did it get to the central nervous system? If you're having an effect on behavior, maybe it needs to get into the central nervous system. Did it get there? If it didn't get there, that could explain why you didn't see a result, and maybe you need to go back and see whether there's a way to make it more bioavailable. If it did get there and you didn't see an effect, did it have the effect on synaptic activity that you hypothesized? Did it have an effect on gene expression, on protein expression that you hypothesized? If it had that effect, but you still don't see the behavioral effect that you expected, then at least you're pointed to, there's something about the connection between these changes at the cellular tissue level and these biological, these phenotypic changes that we don't understand, that we need to go back and learn more about. So you've learned something. I think more than if you go in without those detailed hypotheses about what's going on and just get a null result. Because it's a lot of work, and it's a lot of time for the researchers, and it's a lot of time for the study participants. So you really want to get as much out of it as you can. So really where this is pointing us to is doing mechanistic research. Mechanistic research on botanicals. What could be easier, right? No problem there. Turmeric. I think Adam touched on turmeric and the complexity of turmeric and the fact that turmeric, curcumin, is a complex product. I'm sure you've all heard of it. I'm pretty sure that the 20 million in sales in the US in 2014 did not all go into curry, especially given the number of things that turmeric is supposed to be, turmeric or curcumin is supposed to be good for. As Adam said, this is one of those areas where people often, in their research reports, are not specific and rigorous in their descriptions of what they're using. So again, it's a complex plant product. The curcuminoids that are thought to be the actives make up less than 10% of the plant itself. There are 3 of them. Sometimes when people talk about curcuminoids, they're using purified curcumin. More often, they're using all 3 curcuminoids that are present in turmeric in various ratios, and they often don't report that. Moreover, in solution, curcumin is not all that stable. It spontaneously degrades to different compounds depending on the solution conditions. And people very often don't check that, so it's not clear what's actually going into the experiment in vivo. So this is not what we're trying to achieve, at least not here at the NIH. So turmeric, not very stable, not very bioavailable. And when you're doing in vitro assays, there are things that go on that you need to be aware of. You can control for a lot of them, but you need to be aware of them. Curcumin binds covalently to sulfhydryl groups in proteins. So both in vivo and in any in vitro assay that involves protein, you're going to need to think about what that's doing to your assay outcome. It interferes with fluorescence between about 400 and 600 nanometers. So if your assay has a fluorescence output, you're going to need to control for that. I don't know why, given the bioavailability, you would be working with 10 micromolar concentrations, but it forms colloids in solution at 10 micromolar and above, and it has been reported to disrupt cell membranes. This is why some people have christened it a pan-assay interfering compound. There are others, and they're not all phytochemicals. This is, I don't think this is in the reference list, but in this you have the paper on the website showing in silico that curcumin, as well as several other very popular phytochemicals, capsaicin, epigallocatechin, gallate, genistein, resveratrol, can intercalate into the lipid bilayer of cell membranes. And they've collated data showing that those 5 compounds can each, most of them either downregulate, the minus signs don't mean no effect, they mean downregulate or upregulate a wide range of different cell membrane proteins. Probably these are not specific interactions. They're probably based on some common mechanism, perhaps their effects on the cell membrane, the lipid bilayer. It doesn't mean they don't have biological activity, but it means that understanding that as a specific ligand receptor type interaction is probably not going to work that well. So that is my whirlwind tour of some of the challenges that are stronger in working with botanical dietary supplements. A couple of slides about general research issues that are not unique to botanicals by any means. So I think what I've been trying to tell you is that to the extent that you can thoroughly understand what you're doing and thoroughly describe it, that facilitates your ability to come back and repeat the work and build on it, and it allows others to do that, which is what really advances our understanding of what's going on. NIH and others are thinking more and more about how, not in exploratory experiments, not when you're just hypothesis generating, not when you're developing methods, but that last experiment that you're actually going to publish that's going to form the basis for your your human subject study design. You try to minimize investigator bias by randomizing whether it's cell cultures or rodents, and by blinding the people who are assessing the outcomes. NIH is asking people to say that they're doing that now in grant applications, or say how they're going to minimize the effects of that inherent and unconscious sometimes bias. There are so many different environmental factors that can alter the outcome of an experiment that you probably need to be thinking about and noting in the methods section of your papers and looking for in methods sections. Everything from, did you verify that your cell line really is what it is? There are a lot of mixed up cell lines out there. What passage number is it? What density did you use? Cells on plastic, if you want to be able to translate the research into in vivo in an animal or a human, there are better models of how cells in vivo behave than cells on a plastic dish. There are now organoid models that a lot of people are using. There are tissues on a chip. All of these seem to translate more frequently than cells grown flat on plastic. How you cage your animals make a huge difference in outcome. Anything that affects the gut microbiome, and we know now that even bedding can affect the gut microbiome, can alter the results. And of course, timing. It's all about timing, right? So, no model is perfect. The thing is to be thoughtful about the question that's being asked and how you're asking it. And be aware, for example, that rodent pancreas is, the beta cells are arranged very differently from those in a human pancreas. So think about that if you're using a rodent model for studies of metabolism of sugars. Small sample sizes and abuse of the p-value can lead to a lot of things being published that then don't replicate. There's talk about setting up a registry analogous to clinicaltrials.gov, but for those final publication pre-human study preclinical experiments, so that you publish before you do the experiment how you calculated your target size, what your analysis plan is, what your primary and secondary outcomes are, so that you're saying where the bullseye is before you get the results. So you can't draw the bullseye around the results after you get them. That's going to get us around some selective reporting and ignoring nulls. And a lot of that is in this talk. It's Open Science Foundation, so it's open access. What's NIH doing to make it easier for people to do this rigorous research? One thing that we're doing is, Adam already talked about, the natural product integrity policy that has been in effect at NCAM since it was NCAM, now at NCCIH, more than a decade since I was there, which was before 2011. And then the carbon centers their mandate is to contribute to developing methods and applying those rigorous methods to research on the effects of botanicals in animal systems. So I'll show you a few things about those. This is actually an outdated version of the ODS webpage about them. There are individual links to all 5 carbon program centers from the ODS webpage. You'll recognize it when you see it. There are 2 flavors of centers: the Innovation and Technology Centers, of which I'll start by telling you about the High Throughput Functional Annotation of Natural Product Center. This is based at University of Texas Southwestern Medical Center. It has collaboration at UC Santa Cruz, and also north of the border in Canada, collaboration across the border. They have a really important component outside the US. What they're doing, I think, is really exciting. And it is orthogonal. They're using orthogonal methods. They're using 3 different methods to look at multiple fractions of complex natural products. The 3 different methods, so they take each fraction. One thing that they do is they do a UPLC-MS method that they've developed that allows them very effectively and with quite a bit of accuracy to determine what the chemical structures are in that complex mixture. They take the same fraction. They use a selected set of gene expression assays to look at its effect on signaling networks in the cells. They are trying to move beyond HeLa cells. I'm just reading their progress report now. They're just about a year and a half into their awards, these centers. And so they are not losing the information as they do these high content, high throughput assays on component interactions. They're retaining the information on how multiple components may be contributing to activity. And so far I've told you it's basically agnostic as to mechanism of action. But what I like is that the third assay that they're using is really completely untargeted. It's a cell phenotype-based assay. So it doesn't have to depend on existing genes or signaling pathways. They're looking at anything that's going to affect cell division rate, cell death, cell shape, nuclear size, nuclear shape, actin cytoskeleton, endoplasmic reticulum. So almost any change. And they have shown that for marine microorganisms, they can use this to identify novel compounds whose mechanisms of action were unknown. And their hypotheses generated through this method for mechanism of action have stood the test verification. But they are now working with the botanical research centers that I'm going to talk about in a minute to move these methods onto the greater complexity of terrestrial botanicals. They are interested in collaborating with people who have material that they would like to run through their assay system. Collaborators do need to bring some of their own funding with them, but that's the link to their website where you can find out about collaborating with them. This, in their own words, is how their assay works, what they're trying to do. This gives you a somewhat more enlarged picture of what their cell phenotype assays look like. This is version 1. They're now on version 2, simply using more outcomes, looking at more outcomes. The other Innovation and Technology Center in the Carbon program is at University of Illinois, Chicago. They're developing a number of methods. One thing that Adam sort of touched on is the importance of stereoisomer, of orientation, absolute configuration of compounds. This is one of the reasons why synthetic compounds may not work the same way as natural compounds, because you usually get a mixture of the diastereomers. They have really developed much more efficient methods to use metal-organic frameworks to determine absolute structures. And a major aim of this center is to find out what resources are already there for natural products researchers, put those together where they're easy to find in sort of a one-stop shop, and figure out what additional resources are needed. This is what their website looks like. Their navigator tool, which you can find up here, has compiled several hundred natural products databases, including structure information, toxicology information, biological activity, plants that the natural products are found in. And you can find all those through the navigator. You can separate out and look just at things that are freely available versus things that are behind a paywall. I said there are 5 carbon centers. These are the 3 botanical centers. I talked a little bit about the center at UIC, Women's Health. The center at Pennington, with collaboration with Rutgers, is doing some really exciting work looking at botanicals and lipid and glucose homeostasis. But I want to show you some data from the center at Icahn Mount Sinai School of Medicine that I think does all the things that I've talked about. This is actually from their prior award, which was a somewhat different type of center. But what they're looking at here is the effects of great products, which they're continuing to do. But here in a transgenic mouse model of Alzheimer's disease, you can see that the mice that get the control chow, they plateau in their ability to find a hidden platform under an opaque liquid, whereas the mice that get the great product, they're a bit better. They're maybe 15 seconds to find the platform as opposed to 30 seconds. I think that's my perspective, but they can find it faster. This correlates with bioavailability of specific metabolites of the great product that are generated by specific components of the gut microbiome. In vivo, when they look at these mouse brains after the experiment is completed, they can see that the mice that got the great product have far fewer amyloid beta plaques in their cortices than the mice that got the control product. And they also have reported, this is Giulio Passinetti's group. You can find the link on our website, and you'll find all their publications. They also report in vitro activity of components of the grape product against both amyloid beta and tau. In summary, I've talked about the importance of understanding in as much detail as you need to get a good sense of what's going on, what molecules are in your botanical, what effect they're having on the biology at a specific level, so that when you do an experiment, you understand why in this mouse model you saw this, but in that mouse model you saw something else. In this tissue you saw this, but oh, you have less of the co-receptor in that other tissue and you see a different activity. The more you understand at a molecular level, I think the more you learn when you go into an in vivo experiment and ask humans to give up their time or animals to participate in your experiments. Some of the resources, the Carbon Program website, the NCCIH integrity policy. This is the link to the NIH rigor website, which talks about the things that NIH is expecting applicants and awardees to do to enhance the rigor with which they're looking at what they're doing experimentally and reporting it. This is a summary of part of what I tried to tell you in very simple terms that I hope you can remember. Do I have time for questions? Joe.
Yeah, just this is not a question. It's just to point something out. Go back to your summary slide.
That one?
Yeah, that very last URL. That's fairly new. It's 2016, perhaps late 2015. Those of us who've been doing this for a long time applauded for years the NCAM and then the NCCIH product integrity policy and wished that it was NIH-wide. That essentially makes the policy NIH-wide, that last URL there. It requires investigators to demonstrate that they know what their research reagents, which includes all their test articles and placebos, are made out of. And so that policy, even though there hasn't been as much fuss made over it as the NCCIH policy, is now NIH-wide, and you're going to have to write a separate section in your grant application detailing how you characterize your research materials.
Your key biological resources, your
Right.
antibodies, your transgenics, your cell lines,
Yeah.
your botanical extract.
I have two comments about St. Johns Wort. I actually teach at herb schools and to professional herbalists about how to read and interpret the scientific literature. And one of my favorite things to do is to take that Weber study on ADHD to groups of herbalists whom I'm teaching how to read the studies. And I pass it around, and they don't even read it. And they say, well, of course it doesn't work for ADHD. We would never use it for ADHD. So sometimes, I think a subject matter expert might benefit some of the studies. My other comment is about the difficulty in standardizing St. Johns Wort. When I was a postdoc, I was having a discussion with the dean of the School of Pharmacy where I was a postdoc. And he was telling me that he had done one of the studies using a standardized extract of St. Johns Wort. And I asked him, well, what did you standardize it to? Because it's very difficult to know. And he said, well, what would you do? And as an herbalist at the time, I said, well, what I would do is I would go out into the field at around June 20th to June 22nd between 10 a.m. and 12 noon on a sunny day. And I would pick the St. Johns Wort and look at how it stained my hands, and I would smell it and I would taste it, and I would know it was good St. Johns Wort. So, and I'm a researcher now, and I do understand the challenges of that. I didn't at the time. But it really is a different way of knowing, and sometimes with these botanicals, I think there's real tension between those things.
I love it. Maybe you should have given this talk, but
No, I don't know any of the rest of it.
but I wonder if you looked at St. John's Wort that was harvested between June 20th and 24th on a sunny day, whatever the exact time period is, and maybe it's moving forward as climate changes. Am I allowed to say that climate changes? how would that chemistry differ from the chemistry before, the chemistry after, the chemistry
That I don't know, but I do know
earlier?
that Ed Smith at Herb
But
Farm
would that- might that be a
actually
way to-
looks at a lot of the constituents and, and found that in fact, that if you pick it at its peak when its constituents are pulled up by the sun, it looks different than it looks other times. What that means, I don't know.
So,
And that's why I'm saying there's such a tension, because I think the people who know one thing don't understand the other thing, and nobody understands it entirely.
well, I wonder whether you could bring it together by doing a chemical analysis of product at different times and combining that with some appropriate biological assay, maybe not actually animal behavior, because that's really not high throughput and takes a lot of
Well, I'll
people
try to put
time.
together a study team and put together a proposal.
I'm not making any promises about funding for that.
Yes, thank you for your presentation. So Adam was talking earlier about the standard reference materials that have been characterized and several more in development. Is the intent then for people who would be testing at the bench, would be to use these standard reference materials? In other words, they would be available just like you would purchase standard reagents or certain cell lines or things like that?
These have all been great questions. Another great question. I think, and I'll let Joe or Adam correct me if they don't like my answer, but I think the idea of the standard reference materials is to allow researchers to test the accuracy, and you don't really need a standard reference material to check precision of your methods. But I think that if you're trying to understand, you're trying to find out the activity of an extract and understand its biological activity, you probably want to use both a bioassay that's appropriate, that's based on your understanding of how it's going to exert its biological effect, with the chemistry, appropriate chemistry for that. Because those are not necessarily designed to have specific bioactivities, as far as I know.
You would not use a standard reference material, a certified reference material, as your experimental test reagent in a human or an animal study.
Too expensive.
They're very expensive materials. The expenses, because the purpose there is they are not meant to be representative of this is what a ginkgo biloba extract should look like, or St. John's, whatever. It's not what it should look like. It's just a representative. These are these chemical entities in this very well-defined matrix with these very precisely measured values. So, the standard for a ginkgo could be 25% of one of those flavonoids. That's not to say the standard reference material will have 25%. It could have 20%. The idea is that it's 20%, you know the actual amount there. You use the standard reference materials to calibrate your instrumentation to verify that your methods, your analyses are giving you the right values back, but you're not buying those to use as the experimental material.
Thank you for that clarification. And then, so I would assume then, if you were going to apply for an NIH grant, that in some of your preliminary data you would show how whatever you were doing in your lab tested out against the standard as part of the rigor?
Right, so...
Right.
So part of that new rigor part is that you have to, if you're using a key biological resource like a botanical preparation, you have to explain how you're going to assess it, characterize it, and how that assessment is validated. How you're going to authenticate the material. So part of that would be showing that you have good chemical assays for both reporting and consistency in your own research. But part of that would absolutely be in your application, showing that it has the expected biological or behavioral activity, or that in some assay that based on the published literature, it would be expected to result in that activity.